What is this right here, Acklin?
A coffin.
Yeah.
Do you think somebody stole the mummy? Yep. No mummy. What is this?
Okay. Well, this is kind of like what you did with your model.
What is that
We
thing?
take the skull.
What is that thing in his mouth? This one's mouth.
That's just where see how they took that?
Yeah.
And then they figure out what the skull looked like. And then they come back and what did they add on top of it?
Some clay?
Clay. And then so they used the clay so that they could figure out what?
What they look like?
You got it. That's just like the model you did.
Yeah. Why can't we see the person's face in this one?
You just can't. They're all wrapped.
But they can't scan.
This one.
That's what
Right here.
Is that one face
The
on there?
woman, the mummy of an Egyptian woman who died
When?
in 2,400 years ago.
What?
Oh, look at these jars.
Did they put? Did they put in that? The big one.
These are jars of grain and food that they put in the burial chamber.
Burial chamber? Why did they put food
Well,
in?
and they stored some of them, but most of the stuff that we have is because we found it in the burial chambers. Because a lot of the other stuff got destroyed
By what?
by people and time.
Can we just go right in?
Let's see.
Have you been here before?
No.
Come on over. It's all interactive here. Wait up here, please. It's all interactive back there.
Okay.
And so activate your cards in the green computers and then go around to activities. At the other drawer, when you check out, you'll get a printout, something like this.
Okay.
There's a science lab, a movie theater. They're doing a show right now on illusions. Quick, here, let me go see
Welcome to Expedition Health.
Okay.
To get started, you'll choose an Expedition buddy.
Who do you want to be your buddy? Leave it in. Continue. Hey. Hey, take your pass. Hey, this way.
The green box is the changing temperature of the side of your hand in the wind. Number an orange box is the changing temperature of the side of your hand that is not in the wind. Not the same, are they? What's happening inside your body when the wind blows on your skin? Within seconds, the arteries and veins under the surface of your skin shrink or constrict to keep in the heat of your warm blood. Less blood flows to your fingertips, and when you get really cold, they can turn blue. Your body does amazing things to keep you going at high elevations. I'd heard that people pee more when they climb to higher elevations. Something called diuresis. I decided to put it to the test. When you get up high, your heart really labors to supply enough oxygen to the rest of your body. To help your heart out, your kidneys start to filter out water from your blood, which concentrates your red blood cells, which means you're...
What one am I? Extra small?
They're called water.
Yep. Some goggles?
Oh, you got a microscope. You know why? And then now when mom comes and says, hey, why do you
There you go.
have a microscope? Then you have to say,
Okay.
I want to do this.
What is DNA?
What makes you you?
Yeah, what makes you you. What are the letters that we're looking for? Do you remember?
No.
You don't remember the letters?
ATCG
Yeah.
or ATC.
DNA extraction. Remove plastic cup from the dispenser.
Now where are all the glass cups? Right here?
I guess. Yeah.
Place the cup into the cup holder.
Okay.
Using the graduated cylinder, measure out 100 milliliters of water and pour it into the plastic cup.
Which one?
Which one's a graduated cylinder? You got it. Good job. You need water. Okay. We want to do how much? 100 milliliters. Need help? Okay, where's the 100?
Here.
Okay. All right. Do we have 100 milliliters?
Yeah?
Okay, pour it in the plastic cup.
Using a large spoon, add 1 scoop of wheat germ to the water.
Okay, use the large one and add 1 scoop of wheat germ. Which one's wheat germ?
Germ? Wheat germ?
Add 1 scoop of wheat germ to the water. Good job.
Now what do?
Add 1 pump of
Add 1 pump of liquid soap and stir for 1 minute. Where's the liquid soap? Yep. 1 pump. Good job. Okay. And stir for 1 minute.
This upper mixture is very thick. This is because the cell membranes are dissolved by the soap, releasing nucleic acids.
Stir it and I'll tell you when to stop, okay?
The DNA makes the solution very sticky.
We're not on the triangle letters. What's it starting now?
What you're supposed to stir with is these. And we'll use the glass rods later on. You'll see what you use that for.
The meat pulverizer treats out the proteins and hold the DNA tightly coiled until
Okay. Can you use the small spoon? And you need 1 spoon of
Do I stop
tenderizer
stir?
and 1 spoon of baking soda.
What? Baking soda first?
It doesn't matter.
I'm gonna do baking soda. Baking soda first. Then what?
1 more.
Or baking soda?
Okay, and then 1 of tenderizer.
2 baking sodas?
No, 2 baking sodas. Yep. 1 tenderizer.
Tenderizer?
Okay, add that in there. Okay, now stir it for another minute.
I've been another minute. Will we get to do all the experiments?
Okay.
What?
Allow the wheat germ to settle into the bottom.
Is that all?
Take about
Now
2
take
to
the
3
spoon out.
minutes.
And then do what?
And then, I guess just sit it down there. Okay, now wait 2 minutes.
Now what do we do?
We're gonna wait 2 minutes so that this settles down to the bottom.
And then what do we do? What do you mean by this?
Once the leaching is set up to the bottom,
Is it back to 8 yet? It hasn't gotten back to 8.
fill the plastic tube about half full. Some of the leaching solid may transfer over. That's okay. But try not to get a lot of the leaching solid in
Okay,
the fan.
can you suck a little bit of liquid off the top? About halfway up. There you go. Good job.
Now what do we do? Is this...
bottle containing alcohol. Dip alcohol down the side.
Now what do I do?
An amount of alcohol.
What do I do with this stuff?
Okay. Hold on.
Try not to mix the two layers. Let the tube sit for a minute or two.
What do I do?
The large and small bubbles
Oh, I see.
appear at the
Okay.
interface between the two layers. This is DNA. It is insoluble in alcohol.
Do I put it in there? Do I put it in there? Do I put it?
Okay.
Now do I put it in
Yeah, I
there?
think now you put it in here.
Yep, squirt it right in.
Okay.
Keep going and fill it up about halfway.
Just get some more
Keep putting
liquid.
more stuff in.
Get some more. Just the liquid on there. No, not all the way.
Right, just the top.
Squeeze it back out. Squeeze it back out. Just get it off the very top. Just like you did before. There you go. Squeeze that in there. Okay, let's do 2 more, just like that. Off the very top. 1. Last one.
2.
Okay, now we want to add some alcohol solution. Can you put some of that in here?
Where?
Right here. Okay, now put it just down the side of the tube. Okay, don't touch it to the... Okay, squeeze it. Okay, that's enough, that's enough, that's enough. There we go.
Then what do we do?
Okay, hit the white button.
Now what do we do?
Okay, do you see where the 2 layers are? Where one layer starts and the other layer
If you
ends
keep
right
swirling
there?
and are careful not to mix the two layers, you will be able to pull out a big
Now what do I do? Do I use this?
Swirl it real slowly right
What's
there.
this?
Yep, right there. Swirl real
wad of
slowly.
DNA
Not all the way to the bottom.
collected on the rod is
Back out
composed
just
of
a little bit.
millions of
What are we going to do with it?
and many of these strands are swirled together to make a
Okay,
large
slowly bring it up. Slowly bring it out.
DNA
Bring it out. Oh,
very
look.
quickly.
Look at that. Look at that wad of DNA you got.
You're going to put them on a glass slide, stain them, and look at them under the microscope. So the trackball will first tell you a little bit about cells, and then just move on from there.
See your own cells. At this bench, you will see your own
Skip that.
cells. Your body is made of trillions of cells.
Get a clean slide from the dispenser. Turn either side knob and a slide is ejected.
Got one side.
Yep.
What do I do with it?
Place the slide into the slide holder cartridge. With the frosted
What does it
side
look
facing up.
like? This?
Using a small water bottle, place 1 drop of water on the target in the middle of your slide.
1 drop of... Whoa, hold on. Yeah, that's water. Okay, go for it. Water. 1 drop. Good job.
Get 1 clean toothpick for yourself.
Toothpick. Pick it up. Here. Here. That one? Okay. Alright, and
Take the toothpick and use the round end to gently scrape the inside of your cheek.
Where?
the inside of your cheek. Right here. Okay, here.
Now what do I do?
Put the round tip of the toothpick into the water drop on your slide. Turn the toothpick back and forth and around in the water to wash out the tiny cells into the water.
Good job.
Now what do I do?
Now throw the toothpick away in the toothpick disposal slot.
Put your slide into the slide dryer until the light goes out. When the light goes out, remove your slide.
The slide dryer fixes or dries the cells onto the glass slide. If the cells were not fixed onto the slide, they would wash off during the subsequent staining and washing procedure.
Okay, the light went out.
Now take
Place
it.
your slide onto the flat part of the metal rack in the wash station.
Right here.
Right where?
Just set it right there.
Right here?
Yep. Good job.
Using the methylene blue dropper bottle, place 2 or 3 drops in the center of your slide and onto the dried cells.
Okay. No, that's water. Methylene blue, right here. Put 2 or 3 drops. Okay.
I decide. I choose 3. Is this my center?
1. Okay, that's plenty. Right. 2, 3. There you go. Okay, cap it off and put it back up there.
Let the cells absorb the stain for 60 seconds. The methylene blue is the dye which stains the different cell components, such as the nucleus, cell membranes, and compartments, a dark color. If you didn't use the methylene blue stain, your cells would appear colorless under the microscope.
Let's see what's on.
Take the slide to make a part. How do I pick it out? To rinse the slide by squirting water over it.
Okay. Can you move the slide down to the end here? Down to the end. So, nope. Yeah, that'll work. Okay. Now squeeze some water on it.
How much?
Just a little bit until it washes it off.
This much?
Yep. Okay, let me see. Can I help? Okay. Like this.
Do we get 2? Do we get 2 of the...
Okay. There you go. Alright.
Now what do I do?
Let the blue water run off and drain.
Okay.
Gently blot the slide on both sides with the blotter, being careful not to remove the cell sample.
Okay. Can you tap it on one side and then the other right there? Okay. Just tap it on one side of there, tap it on the other. Okay, there you go. Good job.
Now you are ready to view the slide using a microscope. Follow the next few steps.
X should click into place over the light.
There it is, right there.